Everyone, this is your week 7 session. So in this session, we'll be talking about introduction to spectroscopy, UV visible and CD spectroscopy, fluorescence spectroscopy, green fluorescent proteins, infrared and Raman spectroscopy. Along with this, we'll also look into the applications of Raman spectroscopy, which are Raman microscopy and Raman crystallography. Then we will practice few MCQs and I'll take your doubts. So let's begin with spectroscopy. So spectroscopy is generally the study of how different forms of electromagnetic radiations interact with atoms and molecules. So now there are different behaviors of light which exist, such as absorption. So light can also get absorbed. In that way, if light rays strike on the surface, then it will get absorbed and it will not get passed to the other end. So it can get absorbed to some level. So its intensity will decrease when it will come out from the another end. The second behavior is emission. That means light can also be emitted from the matter. Third behavior is transmission. That means light can completely pass through a substance without getting absorbed. So in this case, its intensity will remain same on both the sides. Then comes reflection, which means that light can strike on the surface and get bounced back. Fifth behavior is refraction, which means that light, when strike on the surface, it bends by some angle and then gets passed out from the another surface. So this means refraction. So this is some of the visualization that how light interacts with matter. Now the devices which capture this thing is known as spectrometers, which are the instruments which measures the absorption and emission of light at specific wavelengths or frequencies. And then comes spectra and molecular fingerprints. So the ultimate aim, why do we need spectrometers, is to check that what is the absorption and emission of light. So each substance has different type of absorption as well as emission spectra. So due to which we can distinguish among different chemical compounds and different biomolecules. So that's why that is the ultimate aim, why we use spectrometry. So spectra and molecular fingerprints. So spectrometers, they generate spectra, which provides information about the atomic and molecular structure of substances. And each substance has unique spectral fingerprints. So that's why we can distinguish among themselves. So here is an example where we have 5 different compounds and each has a unique spectra. So with which we can distinguish among them. So that is the advantage of spectroscopy. And now this is to clear the doubt among the spectrometer and spectrophotometers. So these two terms are mostly used and there is a very little difference among both of them. So when we say spectrometer, it is a broader term and spectrophotometers come under spectrometer. So in spectrometer, we measure the intensity of light, whereas in spectrophotometer, we check that how much light is absorbed or transmitted at specific wavelength. So in intensity, then spectrometer, we'll check it for the range of wavelengths, whereas in case of spectrophotometer, we check it for the specific wavelength. So in this case, we fix the wavelength. So that is one of the major difference. And in later slides, when we will look into the UV visible spectrometers, then we'll see that in case of UV visible spectroscopy, we use the devices which are known as spectrophotometers. And similarly, in IR also, we use spectrophotometers, whereas the spectroscopy such as mass spectroscopy, Raman spectroscopy, fluorescence spectroscopy, and NMR spectroscopy, in these scenarios, we use spectrometers. So this is the how they originally looks. So now we'll talk a little about the history of spectroscopy. So Isaac Newton, he has contributed to the spectroscopy. So in 17th century, he demonstrated that a prism can split a white light into the spectrum of distinct colors. So these are 7 distinct colors arising from a single white light. So this discovery laid the foundation for studying light and its interaction with matter. Later on, Isaac Newton and other scientists such as, Albert Einstein and James Clerk Maxwell, so they came up and they mentioned that this particular light, it has two natures. It has a dual nature, which means it has both wave as well as particle nature. And then after all these things were studied, so it was realized that there are two kind of lines which exist. One is the absorption line and another one is the emission line. So they observed that dark and bright lines in the spectra, which was unique for the chemical elements. So with which, by looking at the spectra, we can distinguish among elements, and these spectral lines could help them to identify the elements. So now in this case, the bright lines which appear, that means there is some kind of vibration, which some kind of light which is reflected, or which means which is given out by the particle. So this is the light emitted, which appear by these light bands in the dark spectra, whereas this spectrum shows the dark lines. So these dark lines appear for the wavelength where the light is absorbed. So these are two different spectra. One is absorption spectra and another one is the emission spectra. So in emission spectra, light lines will represent the wavelengths at which the light is released or emitted, whereas in the absorption spectra, dark lines represents the wavelength of light which is absorbed. So in this case, as it is absorbing the light to move from level 1 to level 2, so in this case, 1 to 2 absorption spectra, so this line represents that wavelength, which is, so wavelength of light which is utilized to show the transition from level 1 to level 2. Similarly, from level 1 it is jumping to level 3. So for that particular wavelength is this one, because we know that there is some energy gap between different levels, and if we supply the energy, supply the wavelength or the light of that particular energy, then only the transition can happen. So similarly, the means the wavelengths, they depends on the how much energy is required. So we know that if wavelength is longer, then that means it has lower energy, and vice versa. So this is about the excitation and absorption, as we have seen. So energy in light excites the atomic and molecular motions, absorbing energy at specific wavelengths, which results in the narrow absorption lines in the spectrum. And excited atoms and molecules later radiate the absorbed energy as light. This explains the presence of bright emission lines in the spectra. So this is how the bright emission lines appear in the spectra. And as we all know, till now we have seen electromagnetic spectra, that there are different radiations which have different wavelengths. And as we move towards the radio waves, these have the highest wavelength and lowest energy. And this is the distribution which shows you that how the wavelength is increasing and energy is decreasing. And the first, this particular area shows you that whether it penetrates the Earth's atmosphere or not. So now we will be focusing on this electromagnetic spectrum when we will look into the spectroscopy. So firstly, we'll talk about the radio waves. So radio waves, they are generally emitted by the radio stations, stars, and gases in space, and which are used in NMR spectroscopy, which we have studied in the last week. Then comes microwaves, which are used in cooking, radar, and communication, and it finds its application in rotational spectroscopy. Then comes infrared waves or infrared rays. So these are associated with the molecular vibrations, and it is used in IR spectroscopy and as well as in Raman spectroscopy also it has applications. Then comes visible light, which is detected by human eyes, and it is involved in electronic transitions in the molecules. Then ultraviolet light, so this causes the electronic transitions in molecules, and that's why it is used in the electronic spectroscopy. So it's also known as electronic spectroscopy because of the transitions it allows. Then comes X-rays. So it allows the transition in the internal electrons, because when we were looking into X-ray crystallography, then we have studied that how a X-ray beam, when it strikes on an atom, then it excites the inner electron, and then that inner electron place is substituted by the electron which is lying in the external orbital. So that is how the X-rays are involved in the transitions of inner electrons. Then comes gamma rays. So these are used in medicines to kill the cancer cells. So remember one thing, that the gamma rays are the rays which have the highest or which has the strongest energy, and their wavelength is shorter. Now, how to develop an electromagnetic wave? So electromagnetic waves, they are generated by the accelerating electric charges. So in this case, this electric charge, it accelerates in both upward and downward direction. And this is how this vibration leads to generation of the wave. So frequency of EM wave, which is the electromagnetic wave, it is equals to frequency of the charged motion. So in this wave, the motion is means of greater frequency, then the EM wave will also have the highest frequency. So when we talk about the visible light waves, so they have frequency of, or you can say they have the wavelength from zero point 0.4 to zero point 0.7 micrometer, which requires high charge acceleration to generate such wavelength, which is approximately zero point 0.5 into 10 to the power minus 6 meters, which is zero point 0.5 micrometer. So EM waves, they consist of perpendicular electric as well as magnetic field components, and this is the propagation of the wave. So wave propagation will always be perpendicular to both electric and magnetic field. So this is the wave equation, which means the wave speed is equals to product of frequency and wavelength. So if your wave speed is high, then you will be able to generate the waves which will have higher wavelength. Yeah, so this is also the example that there is 3 meters per wave and 2 waves per second, so which is representing the frequency, and this 3 meter per wave is representing the wavelength. So as we have studied in the previous session also, so wavelength tells you the distance between two adjacent points which are in the same plane. So for example, two crests or two troughs. So now this is a frequency calculation for visible light. So if our aim is to reach to a wavelength of 0.5 into 10 raised to power minus 6 meters, which we have seen here, that because it is ranged between zero point 0.4 to zero point 0.7 micrometer, so we have fixed it to 0.5, which is in the middle. So for that reason, we require a frequency of 6 into 10 raised to power 14 hertz, so which is a very high frequency. And such high frequencies occur in atomic and molecular regions where electrons move rapidly around the nuclei. So now how can we develop electromagnetic wave artificially? So in that case, there is a mechanism which is known as antenna mechanism, in which the AC power supply is provided, which generates accelerating electrons. So whenever this power supply is provided, then it accelerates the electrons in the antenna arms. Now, due to the acceleration of these electrons, because their frequency or their vibration force will increase, so now electrons will move up and down, producing an outward moving EM field. Because we have seen in the first slide that as the charged particle moves, it will generate some waves. So similarly, after supplying the AC power, electrons, they are continuously moving either in the downward direction or in the upward direction. So due to this quick movement, there will be generation of the EM waves outward. So as the AC voltage changes the polarity, the electron flow and the electric field direction also reverses. So each time, for example, in this case, the supply is towards the downward direction, and in this case, supply is such that it is moving towards upward direction. So it depends on the polarity. So cathode and anode, so respectively, the electron will move towards the positive arm of the AC power source. So similarly, in this case, electron is changing its directionality, due to which multiple such waves could be generated. And ultimately, these waves will be received by the receiving antenna. So this is how the waves, electromagnetic waves are generated by providing the AC power, and it is based on the vibration of the electrons. So now we'll look deeper into the interactions of molecules with the electromagnetic radiations. So molecular interactions with electromagnetic radiations. So if we want to study any molecule, then the first thing is that that particular molecule should interact with EM radiations. If it is not interacting with EM radiations, then we cannot study that molecule. So molecule must exhibit an electric or magnetic effect to interact with the electromagnetic radiations. So as we have seen in NMR, that how the spin changes its directionality. So nuclear spins have magnetic dipoles which are aligned with or against an external magnetic field, and radio frequency energy causes the transition between the spin states. So if no magnetic field is applied, then in such scenarios, the spins are randomly oriented. So these are the nuclear spins. But as we supply the magnetic field, then these spins, they get they get proper directions. So with the help of external magnetic field, because these spins are now properly oriented, so there will be two states which will be generated, and there will be some energy gap between these two states. So in NMR spectroscopy, we have seen that that particular energy to move from one spin state to another spin state is provided by the radio frequency waves. So that is the principle of NMR spectroscopy. So this tells you that the molecule which we want to study, it should interact with these EM waves, then only we can study it. So in NMR spectroscopy also, we have seen that there are some atoms which are NMR active or others which are NMR inactive. So that is the main precondition for studying the electromagnetic behavior of the molecules with the electromagnetic radiations. Then comes rotational spectroscopy, which is lying in the microwave region. So in this case, we use microwaves. So molecules with net electric dipole moment, such as water, they experience the dipole direction changes due to the rotational motion. So this is how, due to rotation, the molecules changes if we provide the microwaves. So this is how, with the help of microwaves, we can study the behavior of molecule. Similarly, vibrational spectroscopy, which includes the infrared radiations. So these are the example of EM waves, that how they interfere with molecules and how we are able to see the changes. So in case of microwaves, we see that there is some rotation in the molecules within them. Then in infrared region, there are some vibrations. So with the help of differing vibrations, we can compare among different molecules. And similarly, in case of electronic transitions, which are mostly observed in the UV region. So in that case, there is absorption and emission of the light. So which is also known as absorption and emission spectroscopy, in which the electrons move between the orbitals, changing the dipole moment of a molecule. And this differences in geometry, size, and orbital arrangement, which will influence these transitions. So one by one, when we'll go into the detail of these spectroscopic techniques, then it will be more clear that how it works. So now we'll talk about spectra and energy transitions. So as we have seen that whenever the white light comes and it passes through prism, then it breaks down into different continuous spectra into multiple such colors. So continuous spectrum means which is formed when the white light is passed through prism and displaying the full range of wavelengths, which means nothing is absorbed, everything is passed as it is. So which means that is continuous spectrum. So that's why it is known as continuous spectrum. Second thing is absorption spectrum. So absorption spectrum occurs when atom, ions or molecules, they absorb specific wavelengths from incoming light, and it is seen as dark lines in the continuous spectrum. So this is the continuous spectrum in which the light of all the wavelengths is present. Now in the case of absorption spectrum, what can happen is that in this case, the example is of hot gas. So if there is hot gas, then there will be some molecules present in between. And as the light is passing through this gas molecules, some molecules might absorb the light of particular wavelength. So in such scenarios, when we will look into the spectra, then in this case, cold gas. So in this case, this cold gas will absorb some of the light which is coming. And in the absorption spectra, this is the continuous spectrum in which there are some dark lines. So these dark lines represents the wavelengths at which that particular wavelength light is absorbed by this cold gas. So that is the difference between the absorption line spectrum and continuous spectrum. So in absorption spectrum, we'll observe some dark lines which will tell us about the absorptions or the wavelength at which the light is absorbed. Similarly, in case of emission spectrum, where the molecules emit the light of specific wavelength, so in this case, we will see bright lines against the dark background. So in this case, the spectra is dark and bright lines only appear for the places for that particular wavelength which is emitted by the molecule. So similarly, if we compare the absorption and emission line spectrum, then we will see that the light which is absorbed in absorption spectra, that will be emitted in the emission spectra. Okay, so the dark lines in the absorption spectra, they appear as bright line in the emission spectra because these radiations have been absorbed and later on they will be emitted. So this is the difference between the absorption spectra and the emission spectra. And then we'll talk about the energy transitions. So transition happens when the energy of incoming radiation matches the energy gap between the states. So del E is equals to h nu, where nu is the frequency and which is equals to hc by lambda, where lambda is the wavelength, c is the speed of light, and h is the constant, Planck's constant. So this ensures that the molecule absorbs a precise wavelength of light. So which wavelength of light is absorbed, it depends that how much energy is required for the transition. So depending on that particular energy gap, only specific wavelengths will be absorbed. Then comes line broadening. So generally, when we observe the spectra, then if particular wavelength is absorbed, then we should see a clear peak. Or in this spectra also, we should see a clear line, but this is not the case. In certain scenarios, you'll find that the absorption lines which appear, they appear as a broadened peak. So there could be multiple factors responsible for this broadened peak, such as temperature and pressure effects. So now we'll check that what are the different factors for this spectral line broadening. So first of the factor is the instrumental factors. So a finite slit width in spectrometers allows the light of slightly different wavelengths to pass. So this is the slit which is allowing the wavelength of light to pass through it. So in certain scenarios, what can happen is if this slit distance is increased. So in this case, previously only a little bit of light is allowed to pass. So which means that there will be only a particular wavelength of light that will be passing. So in that case, the possibility of observing a sharp spectra is higher. But in the cases where the slit width is large, so now the multiple wavelength light could be easily passed through the slit. So in this way, when we will observe the peaks, so because of the contribution from multiple wavelengths, this peak will be broadened. Similarly, in this case, the distance between the slit is quite high as compared to this one. So due to which the multi-wavelength light is passing, and now all these wavelengths are combining together, and now the peak which will be formed, it is the most broad peak. So this is the reason, this is the instrumental factor, that it is depending on the slit width, that how much light you're allowing to pass. So if width will be high, then multi-wavelength light will be able to pass, due to which the spectral lines will get broadened. And this results in the symmetrical broadened absorption peaks around the expected wavelength, which is equals to lambda is equals to lambda t. Now, next reason is the sample factors, which means that molecules in the liquid or gas, they are in the constant motion and they are colliding with one another. So collisions, they will affect the vibrational and rotational motions, which will cause the spectral broadening, and which is also known as Doppler broadening, which means that the moving molecules, they experience the frequency shifts in absorption due to the Doppler effect, and faster moving molecules leads to broader absorption lines. So in this case, when the gas particles are rest, so in this case, this is the emission lines. So wherever the light is emitted, those lines appear as a bright lines. So the lines are quite clear. They are sharp. Whereas if these gas particles are continuously moving, then that means there are certain vibrations which are undergoing. So now they will lead to the broader lines. So this is known as the Doppler broadening, in which the moving molecules brings about the change in the frequency due to the absorptions. The third reason is the intrinsic or natural broadening. So according to Heisenberg's uncertainty principle, del E into del T is always greater than or equal to h prime by 2, where h prime is h by 2 pi and h is the Planck's constant. So which states that a shorter excited state lifetime results in the greater energy uncertainty. So if there is uncertainty in the energy, so which will leads to a broader peak, and large del E leads to a broader spectral lines, while longer lived states have sharper peaks. So in this case, as you can see, this is the excited state and this is the ground state. So ground state always have the small del E, which means small energy gap. So as this del E increases, the broadening of peak also increases. So in this excited state, because of large del E, and del E and del H are also dependent on one another. So excited state has a larger del E, which means it has shorter lifetime, and shorter lifetime, which means it has the higher value of this del H, or you can say H prime. So due to which it will lead to the broadening of the peaks. So this is the intrinsic broadening, and these are the factors which leads to the broadened peaks. Now, before reading about the UV spectroscopy, firstly we'll look into the basic concepts of spectrometry. So first thing is spectrometer, so which is the device which tells you that how much light is absorbed and what is the intensity of the that particular light when it is emitted. UV visible spectrometers, they operate in 190 to 800 nanometer range. So this is the wavelength range for UV or visible spectrometers. Now there are different components of the spectrometer. First of all, light source. So there is a requirement of a light source which will supply the UV or visible radiations. The second thing is the dispersive element, which is also known as the monochromator. So monochromator allows a specific wavelength of light to pass through it. So keep one thing in mind, which we have previously also discussed, that the spectrometer is a big term and spectrophotometer is a small term which comes under the spectrometer. So whenever we'll talk about the UV visible spectroscopy, then we will talk about the spectrophotometers, which checks that how much light is absorbed. So it is based on the light absorption at a specific wavelength. So similarly, in spectrophotometer, we require something which can allow us to pass a specific wavelength of light through our sample. So for that reason, we have a monochromator in case of spectrophotometers. So monochromator, it, as you can see, the light source is passing the light. Collimator is allowing a proper, it's providing the proper direction. So the lights in only one direction will be allowed to pass through the collimator. Now it will enter through prism. Prism will split the light into 7 individual components. So now there is a slit, which is a wavelength selector. So this entire three things, these together work as a monochromator. Now this wavelength selector, it will only allow a specific wavelength of light to pass through it. Now that particular light will have some intensity I naught, which will pass through our sample. Now, this sample contains certain molecules which have the ability to absorb the light because the spectrophotometry is based on the absorption of light. So, in UV spectroscopy, we will be only focusing on those molecules which can absorb the light. So, similarly, now it will absorb some light, and I represents the intensity of light which is passed after the light passed through the sample. And ultimately, the detector, which is the photocell in this case, it will record the intensity. It will tell you that how much is the absorption at this particular point. So more the absorption, more will be the concentration of the molecules in the sample. So later on, we'll see what are the different equations and how we do it. So this is just a brief overview that what are the components of spectrophotometer. Now there are certain key equations which must be kept in mind. So first thing is transmittance. So as the name suggests, transmittance. That means something which is transmitted from one surface to another surface. So in this case, transmittance is equals to I by I naught, which means the intensity which is transmitted. So this is the intensity which is passed through the sample and which is in the end which is obtained. That is the intensity of light represented by I divided by I naught, which means what is the intensity of light before passing through the sample, which is I naught, which is also known as initial light intensity. So if transmittance equals to 1, which means nothing is absorbed, the light intensity is exactly identical. But this is not the case in UV spectroscopy because the sample which we use, it undergoes the absorbance of light. So you'll find that transmittance value will always be less than 1 in case of UV visible spectroscopy. So then comes the another term which is absorbance, which tells you how much light is absorbed. So it is calculated by, it is the negative log of transmittance, which means negative log of I by I naught, and which shows you that if there is higher absorbance, means the greater light absorbed by the sample. Now there are certain equations which are related to this absorbance measurements. So that is known as Lambert's law. So Lambert's law states that light intensity decreases exponentially with increasing the path length in the absorbing medium. So for example, in this case, as we have seen, so this is the sample which is present in a container. So this is a qubit in this case. So it has some distance. So this path length from where the light is traveling. So how much distance the light has to cover when it is passing through the sample, that is known as path length. So if we increase the path length, for example, instead of using a qubit, if I'm using a bigger beaker, so in that case, I'm increasing the path length. So what my Lambert law is saying, it is saying that if I'm increasing the path length, then the light intensity will decrease. Why it will decrease? Because now the path length is increased. So now the sample is allowed to absorb more of the light. So that's why intensity will decrease. Now, the second law is the Beer's law, which states that light intensity decreases exponentially with increasing the concentration of absorbing medium. So, with length also it's increasing, and with the means if we increase the concentration, which means if the molecular concentration will be high, then it will. So, if more molecules are present, then the absorption will also be higher. So, if more light is absorbed, then its intensity will eventually decrease when it will be passed from the after passing through the sample. So that is what Lambert's and Beer's law is all about. So now we have combined both the laws when we study spectroscopy. So the combined law is known as Lambert-Beer's law. So which states a linear relationship between the absorbance, concentration, as well as the path length. So in this case, the equation is A is equals to ALC, where capital A represents the absorbance, small a represents the molar absorptivity, which is also known as extinction coefficient in some cases. And in many places, you'll see that instead of writing A, they have written it as epsilon. So there could be multiple ways to represent these formulas. And this unit of molar absorptivity is liter per mole centimeter, which depends on the wavelength and the substance. Then L is the path length, which is represented in centimeters, which means the distance that the light travels when it passes through the sample. And C means the concentration of the analyte, which is calculated in mole per liter. So this is about the Lambert-Beer's law. Yeah, so this is the representation that how the increase in concentration as well as path length leads to decrease in the intensity of light, which is ultimately reaching. And with the help of this, we can also identify that if at what particular concentration, what will be the value of absorbance. So for example, if you have the unknown compound in your solution, So in that case, by looking at the absorbance, you can plot the concentration. So you can find out the concentration of the unknown material by using the UV visible spectroscopy. And second thing is the fundamental in UV visible spectrometry and quantitative analysis in the chemistry and biology. So mostly it is used for the quantitative analysis. Okay, now we'll look into the UV visible spectroscopy and what are the major classes of electron transitions which happens. So because we have studied that in UV, it is also known as the electronic spectroscopy because in this, the transitions are happening at the level of electrons. So now what are the different types of transitions which could happen? So if we have the two p orbitals, then there are two scenarios which are possible. So first scenario is if there is head-on overlap between these two p orbitals, so this will lead to the sigma orbital or which is known as sigma bonding orbital because there is some kind of addition which is happening. If they are in phase, then they will lead to the formation of this sigma bonding orbital. If they combine out of phase, which means this positive is matching to this negative, and then means if there is out of phase means addition of both of them, so they will cancel out each other. So in such scenarios, sigma star anti-bonding orbital is formed, and there is some energy which is required for the transition from sigma to sigma star orbital. Now the second possibility of second transition which can happen is, so if two p orbitals, instead of joining head to head, if they laterally combine, so there is parallel combination of two orbitals. So in such cases, there will be generation of the pi orbitals. So first will be the pi bonding orbital if they are combining in phase, and then there is another orbital which could be formed as the pi star. So in case of pi star, this is known as anti-bonding orbital if the combination is out of phase. And for their transition, moving from pi to pi star, that transition is known as pi to pi star transition. So these are the two different types of transitions which are observed in UV visible spectroscopy. And this is how these bonding and anti-bonding states are formed. And then there are another orbitals which are known as non-bonding orbitals, which arises due to the lone pair of electrons. So now the stronger, if there is transition from sigma to sigma star, then that is known as strong absorption. So which generally observed in the single bonds. So if there are single bonds, then you will see this type of transition, and it requires high energy of UV radiations. So which means that if you require high energy, then that means you have to absorb a stronger, means this absorption strength will also be high because high energy is required. So if there is transition of sigma to sigma star, then you will find that the absorption will be quite high. Then comes N to sigma star, which means from non-bonding to sigma star transition. So this is the this requires the moderate energy. So you will see that there will be a moderate absorption values which you will observe, which involves the non-bonding electrons. For example, lone pairs in oxygen, nitrogen, sulfur, and halogens, and it is observed in the saturated compounds with the heteroatoms. Then comes pi to pi star transition, which is common in the organic compounds, and it occurs in the molecules with double or triple bonds. So pi bonding orbital will only be formed if there are some double bond present. So in double bond, there is always a sigma orbital as well as a pi orbital. And if there are so if there is a triple bond, then there will be two pi bonds and one sigma bond, which is present between the two atoms. So similarly, it is observed in those organic molecules which contains either double bonds or triple bonds, and this is a low energy transition. So in this case, the adoption will not be that intense as in the case of sigma to sigma star, and this is more frequently observed. So in UV visible spectroscopy, this kind of transitions are majorly observed. And then comes N2 pi star. So this is also means the moderate absorption, and it involves lone pair of electrons interacting with the pi star orbitals, and it is common in the compounds with, for example, if there is a carbonyl bond or if there is a C double bond N or N double bond N. So this is how different electronic transitions are taking place, and this is the principle of UV visible spectroscopy. So different compounds have different bonds, so they can show different transitions, and based on those transitions, there will be different absorptions of light. So with different absorption values, we can find out that what kind of organic compound or what kind of biomolecule is present in our sample. So now how we will check the UV visible spectroscopy in proteins? So for that, we must understand that which are the amino acids which shows this absorbance. So most proteins show an absorbance maxima at 280 nanometers. So which means at this particular wavelength only, the proteins will absorb the light. Whereas nucleic acids absorb the light at 260 nanometers. So these molecules, they require some kind of energy for the transitions. And that energy is also dependent on the wavelength because wavelength and energy are related to one another. So 280 nanometers and 260 nanometers. So this represents the wavelength of light, which is absorbed by these molecules. Now, aromatic amino acids such as tryptophan, tyrosine, and phenylalanine, they are responsible for the absorbance at 280 nanometers. So, if, for example, if I have a protein which do not have any of these three amino acids, then that will not show any absorbance. So, in that case, the intensity of light, whatever intensity is the light which is entering the sample, it will exit with the same intensity. So, these are the three key amino acids which are required for the absorbance. And then interference from nucleic acids. So many cases, what can happen is that you are taking a protein sample, it could have the impurities. So if there are nucleic acid impurities, then you will see that whenever you are observing the spectra, then there might be some absorbance which is also happening at the level of 260 nanometers. So which can contaminate our calculations. So to correct our formula, to get the correct concentration of protein, this is the formula in which 1.55 into the absorption at 280 nanometers minus 0.76 into absorption at 260 nanometers. So this is the correction. So after correcting it for any of the nucleic acids which might be present in our sample, we will find the concentration of our protein. Then comes spectral analysis of these aromatic residues. So if we compare among these three residues, then tryptophan is the one which has the strongest absorption in proteins due to its large conjugated system. So now why these amino acids only showing this behavior? Because there is a conjugated system which is present. So in all of them, these are the aromatic amino acids. So due to presence of this conjugated system only, there is a possibility of transitions. So with the help of these double bonds and all these things, they are present. So that's why the electronic transitions can happen in these amino acids. And that's why they show the absorbance of UV or visible light so that they can undergo transitions. So tryptophan is the best uh, amino acid which shows the highest absorption. Then comes tyrosine, which shows the intermediate absorbance. And then comes phenylalanine, which shows the weakest absorbance. So now UV spectroscopy is complete. Now the next thing is the circular dichroism and polarization of the electromagnetic radiations. So CD spectroscopy, it is also based on the absorption technique. But in this case, in the previous case, we were looking at the absorbance of a particular wavelength of light. So in this case, we will see that it's not based on the wavelength absorption, but it is, although it is based on the absorption of light. So in this case, it relies on a differential absorption of left and right circularly polarized light. So in circular dichroism, there is a device which is known as a polarizer, which is used, which polarizes the light into one plane. And then this particular light is having the two components. So there is a right circular polarized light and a left circular polarized light. So depending on differential absorption of either one of these lights, the molecules vary among one another. So now we'll just quickly go through this. So first of all, let's talk about the types of EM radiations. So first of all, there is a unpolarized EM radiations, which means that the wave is composed of the randomly oriented electric field vectors. So as we know in the EM wave, we have both the electric field as well as the magnetic field, and this is the propagation. So light is passing in all these directions, and there is no proper plane of this particular light. It is randomly present, randomly oriented. These vectors are randomly oriented. So polarizer is a device. What it does is it selectively allows the light waves of a specific polarization to pass through it, and it will block the other light. So, for example, it will only allow the light which is passing in the Z plane. Only that will be passed. And the Y plane and the X plane will not be allowed to pass. That is the work of polarizer. Plane polarized EMR, that means plane polarized EM radiation. So after passing through a polarizer, only one plane of polarization is transmitted. So that is known as the polarization. And what is the importance of CD spectroscopy? So it requires, first of all, it requires the circularly polarized light, which is generated from a plane polarized light. So because in this case there are two components, electric field component, magnetic field component. Now we will only allow the one component to pass. For example, we only allow the electric field component to pass. So in the next slide, we'll see what a circular polarizer does. So it will allow only one plane of light to pass, and then the circular polarized light will also have two different phases. First will be the clockwise or right circular polarized light. Another component will be the anti-clockwise or the left circularly polarized light. So depending on the variable absorption of either of these two lights, different proteins secondary structures could be identified because each secondary structure has the unique absorption for these two circularly polarized light. Yeah, so this is just a representation. In later slides, we will see it in detail. So now I'll tell you about the type of polarization. So first thing is the linear polarization, which is also known as plane polarization, which means if I have two components, then I only allow the light in one phase to pass through it or in single plane. So for example, in 3D, I have XYZ plane, so I'll only allow it to pass in Z plane only, for example. Then comes circular polarization, which is observed in the CD spectroscopy. So which means the electromagnetic field maintains a constant magnitudes but rotate at constant rate in plane perpendicular to the propagation direction. So in this case, it will be it will have the components, circular components, which have the circular vectors in all the directions, and it will be in both the perpendicular planes, but the magnitude, that means the amplitudes, or you can say the crest and the troughs, they will be exactly same, means they will not vary. So which means that it will have a constant magnitude and it will be means perpendicular to each other. So both the directions were perpendicular to each other. So that is known as circular polarization. And then comes elliptical polarization. So a general case where both the magnitudes and orientation can vary. So for in this case, so they can means, for example, in circular polarization, we are making sure that they should be perpendicular to each other. So in this case, they cannot be perpendicular also, and they can also vary in their amplitudes. For example, the crest of this might not be same for the other wave which is passing. So that is known as the elliptical polarization, in which the waves which are passing or the components which are passing, they can vary in their orientation as well as the means magnitudes. Yeah, so this will focus on the circular polarized light. So circular polarized waves, the electric field vectors rotates while maintaining the constant magnitude. So in this, the vector is rotating in all the directions, but at each circle, you'll see whenever it completes the circle, the magnitudes remain the same. So magnitude is the peaks, or you can say the amplitude remains the same for all the waves. So there are two possible orientations. It can either be right-handed or left-handed. So right-handed is, or you can say when it is clockwise, so that is known as RHCP, which is right-handed circularly polarized light, where the electric field rotates clockwise when viewed in the direction of propagation. So this Z represents the direction of propagation, whereas left-handed circularly polarized light means when the electric field rotates counterclockwise, or you can say anti-clockwise when we are looking at with reference to the direction of propagation. So these are two different types of circularly polarized light. Yeah, and CD spectroscopy is based on the differential absorption of these two. Yeah, so this is an example. For example, a light beam is passing, so it will have an equal contribution from both the right as well as left-handed circularly polarized light. Now, if I keep some compound which is optically active in middle, so each compound has different, you can say it can absorb these two lights differentially. For example, in this case, it has a preferential absorption for my right-handed circular polarized light. So in this case, it will absorb my right-handed circular polarized light. Now, if I compare among both these components, so I'll see there will be a decrease in the right-handed circular polarized light, whereas my left-handed will increase. So which means there is the preferential absorption of right-handed polarized light. So which will tell us the difference among different optically active compounds because they vary among their absorption among both of these. Means depending on the preference, they will absorb the right and left-handed circularly polarized light variably. So this is the principle of CD spectroscopy. This is how we can differentiate among different molecules. And now how to measure the CD spectra. So after interaction with the chiral sample, left and right circularly polarized light have different amplitudes. Because what we have seen, what a circular polarized light means, that it has the same amplitude for both the components, both the right and the left hand circular polarized waves that will have the same amplitude, but after it interacts with something, it will have a different amplitudes. So the resulting light is the elliptically polarized, and the ellipticity is what CD measures. So what we have seen in the previous slide, that there are three types of polarizations. So in circular polarization, the amplitude remains fixed, the magnitude is constant. But in elliptical light, the amplitudes vary, the orientation can also vary. So what a optically active molecule is doing, it is converting this circularly polarized light into the elliptically polarized light, where the amplitudes are varying. So my CD spectrophotometer, which I am using, CD spectrometer, it will check that how much ellipticity is happening. So it is based on the calculation of that ellipticity. So CD signal is given by del A, which means change in absorbance at particular wavelength, is equals to AR, that means the absorbance of right-handed circular polarized light at that wavelength, minus AL, which is the absorbance at the left absorbance of left-handed circularly polarized light at particular wavelength, which can also be written as epsilon r lambda minus epsilon n lambda LC. So this is where we are putting the Beer-Lambert's law. So according to Beer-Lambert's law, we have seen that absorbance is equals to epsilon CL. So where epsilon was the absorbance or the molar absorptivity. And in this case, because we are finding the difference among the absorptivity of both the right and left-handed circularly polarized light. So in this case, this is referred to as del E lambda, which is the difference in the extinction coefficients. So this particular E, which was previously small a, so this can be called as molar absorptivity, extinction coefficient. So these are two names by which we call it. And del A is the absorption differences, L is the path length, and C is the sample concentration. So this is how this equation is derived. So it is because we are putting the values from the Beer-Lambert's law. So CD signals are the small differences between the large values. So for example, if we have del E, which is equals to 3 mole inverse centimeter inverse, whereas epsilon is 6,000 meter inverse centimeter inverse, mole inverse centimeter inverse. So which means that the absorptivity of a solution is so high. So the difference which is coming out, that is very small. So that's why we require very sensitive instrument which can measure this small level of difference, because this is just 0.05%. So this is how CD spectroscopy gives us results. And this is also one of the examples. So when we look for this absorption spectra, so we will look for the wavelength and the light absorbed. But in case of CD spectra, we compare the wavelength versus this del E, which is the change in the absorption, or you can say the change in the extinction coefficients. So this is how both of the spectra varies, absorption as well as CD spectra. And different molecules, they also vary among, means they will also show variations in the values of this wavelength versus del E. So that is how we differentiate among different molecules. So now we'll take the example of protein secondary structures. So CD detects the interaction between the transition dipoles of peptide bonds. The relative orientation of these dipole changes with secondary structure. So the main principle of the spectroscopy is that in any EM wave, it should bring about any change in the atom. Then only we'll be able to study. So in case of CD spectroscopy, the change which is happening, that is happening due to the orientation of these dipole moments. So if there is change in the relative orientation, then different CD spectra will be obtained. And what is mentioned here is that different protein secondary structures such as alpha helices, beta sheets, beta turns, they have different orientation of these dipole moments. So due to which there will be change in the absorption values, and different peaks or different spectra will be observed. So the far UV region, which is less than 240 nanometers, is the most useful for analyzing the secondary structures as it reflects the peptide bond absorption. So this is the example in which polyl lysine is considered. And this particular peptide, which is composed of multiple lysines joined together. So when this was compared, so it was observed that when this particular peptide is absorbing the, when this peptide is adopting the alpha helical secondary structure. So in that case, the molar ellipticity value, first peak was observed at 193 nanometers, and then it has two negative peaks at 208 and 222 nanometers. In case of beta sheet, it has a positive peak at 195 nanometers and negative peak at 218 nanometers. Whereas if it adopts the random coil structure, then it has a negative peak at 195 nanometers. So which means that a peptide or a protein, it shows different peaks for different secondary structures. So with this, by looking at the CD spectra, we can predict that what would be the secondary structure my protein is adopting. And this is another table which tells you where the negative band and where the positive band will be obtained. So based on this, we can, we can say we can annotate our spectra, and we can identify that what are the secondary structures present in the protein which we are considering. Yeah, so this is from another paper which is showing you that how the free state and the bound state protein can also vary. So they can also show different del E values with respect to wavelengths, with which we can also find out that whether the protein which I'm considering, whether it is in bound state or free state, and what are the different components, means how many alpha helices are present, how many beta sheets are present, or whether it has some random coil structure, whether it is disordered. So all these calculations can be performed by looking at the CD spectra. Yeah, and this is also another relationship which tells you that how with increase in temperature, because with increase in temperature, the secondary structure which was present, now the unfolding can take place because temperature is increasing, so bond breakage can happen. So with increase in temperature, the fraction of unfolded protein is increasing. So this is the example which shows you that if the protein is at 25 degrees centigrade, then it has this particular spectra is obtained for the alpha helical structure. So in this case, the protein which is selected, it has the alpha helical region which is present. So at 25 degrees centigrade, this shows proper values of this particular phi, which represents the del E or del epsilon. So in this case, as we increase the temperature, this alpha from alpha helical CD spectra, it is moving towards more towards the random coil peaks, where only one negative peak is present and no positive peak is present. So this shows that with increase in temperature, the secondary structures can deform because due to unfolding, the number of alpha helical content which was previously present, that can decrease. So from alpha helical structure, it is moving more towards the random coil orientation. So this is the relationship that how we can study in real time with the help of CD spectra, that how the temperature is influencing the secondary structure of a protein. And this is the another example in which, for example, if there is more alpha helical content, if there is less alpha helical content, then how we can distinguish among them. So this is how different spectra is observed for different proteins based on their alpha helical content. So if you're interested, you can go through this. And similarly, this is another example. So in this case, with the help of CD spectroscopy, we can also calculate the percentage of secondary structures by fitting the unknown curve, which is theta u, to a combination of standard curves. So according to the simple case, what is the equation? So theta t is equals to x alpha theta alpha plus x beta theta beta plus x c theta c. So which means alpha helix, beta sheets, and coils. So which vary. So x alpha, x beta, and x c, they tell you that what is the percentage of alpha helices, beta sheets, or random coils which are present in the sample. So we can means check for that. So if we have already predicted percentage of all of them, then by looking at the CD spectra, we can fit the data and we'll check that whether the calculation is right or not. So for example, in this case, they have studied it for the myoglobin. So in this myoglobin protein, there is 78% helix which is present and 22% coil is present. So when we will fit the data, so it was observed after putting the values in this, it was observed that X alpha was 80%, X beta was 0%, and XC was 20%. So which means that it agrees well with the secondary structural calculations from the high resolution structure. So when X-ray crystallography or other high resolution techniques were performed, then 78% helix content was identified and 22% coil was identified. So similarly, by using the CD also, similar results were obtained. So which means that CD spectra can also tell you about the concentration of, or you can say about the amount of how many alpha helical, beta sheets, and other coils are present in the protein. Yeah, and this is the another case. So there is a molecule which is the trifluoroethanol. So this is the molecule which is responsible for inducing the helicity in all peptides. So in coiled coil structure, TFE breaks down the helical diamonds into the single helices. So what do we mean by induces helicity? Which means it increases the alpha helical content. And how does it increase? So for example, if I have a coiled coil interaction in which the two alpha helices are joined to one another. So in such structures, what this TFE is doing, it is splitting it into two individual alpha helices. So which means it is increasing the number of alpha helices. So from dimer state, it is increasing it into the monomer state. And although the secondary structure remains the same, CD spectra show the changes due to the environmental effects. And these are some other applications of TFE, that what are the different things which we can observe by providing TFE. Yeah, and in this also it is showing that as I am increasing the concentration of my TFE, how the del or how the theta is changing. So del epsilon, or you can say how the ellipticity values are changing with respect to wavelength. So it is clear that as the TFE percentage is increasing, it is moving to the more alpha helical content. So means more prominent peaks are observed. Or for example, in this case also, the similar thing is shown that with increase in the percentage of TFE, my alpha helical content is increasing with respect to my previous protein alpha helical content. And with respect to temperature, and this was with respect to wavelength, because as we know that if you increase the temperature, then the because we have seen in the previous example also, if we increase the temperature, then the means secondary structure begins to break down. So protein becomes more unfolded. So in such scenarios, with increase in temperature, we can see that there is decrease in the peak values which are observed. So which is for all the cases. But if we individually compare them, then we'll see the highest peak value is obtained where the TFE concentration is high for the alpha helical band. And there are certain limitations of the CD analysis. So first limitation is the oversimplification, uh, which means that because the CD spectra, it is completely based on the secondary structure estimation, or it is only telling you about the secondary structure. But what is actually happening is that there are some tertiary structure which is also present. So there are some other bonds which are also responsible for judging it. So secondary structure identification is not enough. There are some other factors which also influence the structure of our protein. So because it is oversimplified and it is only looking for the certain secondary structures such as alpha helices, beta sheets, and random coils. But what about other secondary structures such as beta turns, coils, or other secondary structures which are also present? So they are not considered. And similarly, tertiary structure influence far UV CD spectra, which are not solely determined by the secondary structure. So by just looking at secondary structure, we are underplaying it. These are the limitations. Now we will move to the fluorescence spectroscopy. So till now we have covered UV visible spectroscopy as well as CD spectroscopy, because in CD also we are using the UV radiations. But now we will talk about the fluorescence spectroscopy. So before understanding the fluorescence spectroscopy, there are certain terms which we need to clear. So there are different ways of relaxation from excited state, and this is the Jablonski diagram which represents it. So now we will study excitation and relaxation pathways. So when a molecule absorbs light, the electron, it moves to the higher excited state. As you can see from S0 state, it can move to S1, S2, S3. So these are higher singlet states, whereas T1, T2, T3, these are the triplet states to which a molecule can means go. So there are some relaxation mechanisms which are present. So first is vibrational relaxation, which means the molecule dissipates energy by conformational changes. So it relaxes to a lowest vibrational level of the excited state. So for example, this is S0 state, this is S1 state. So if my molecule is moving to S1 state, or my electron is transitioning to S1 state by gaining some energy, so now this S1 state has different vibration levels. That means it is not changing its level, or these energy levels are not changed, but it is for a single energy level. There are some vibrational levels which are present. So when my molecule is relaxing or is moving among these vibrational levels, that is known as vibrational relaxation. Then comes fluorescence. Fluorescence means when it is moving from the my S1 energy level to S0 energy level, and it will release some kind of energy in the form of fluorescence. So if the molecule cannot vibrationally relax to ground state, it emits the light. So in this case, light is emitted. So this green arrow represents the fluorescence, where the movement is happening from one energy level to another energy level, or from higher energy level to lower energy level. This is known as fluorescence. And when light is released, if there will be vibrational relaxation, then some light or some means energy will be dissipated as heat. So whenever from this state it will enter to this state, so it's means the light intensity which it will emit, that will be less. So in later slides, we will study that also. Then comes inter-system crossing. So inter-system crossing means that when it moves from one state to another state. So from singlet state to triplet state. So this is known as change in the systems. So singlet to triplet movement is interstate movement, which is known as inter-system crossing. So if an excited singlet state overlaps with the triplet state, the molecule crosses over into a triplet state. This is known as intersystem crossing. Then there is another term which is phosphorescence, which means when it is relaxing from T1 to S0 state. So that is one difference between the fluorescence and phosphorescence. So in fluorescence, we are moving from singlet to singlet ground state, but in phosphorescence, we are moving from triplet state to singlet ground state. So this is the difference, and these are the different relaxation mechanisms. So in fluorescence, we will be looking into this particular relaxation mechanism, where from a higher singlet state, my molecule is relaxing back to a ground state by releasing some energy or some light of certain wavelength, which will be captured. And this is the principle of fluorescence spectroscopy. And this diagram, which is telling about different relaxation levels, is known as Jablonski diagram. So now the concept which I was discussing previously is known as Stokes shift. So what Stokes shift means? So this is the Stokes shift is the energy gap between the maximum absorption and maximum fluorescence emission. So for example, if some light is absorbed, then that light has to be emitted. But there is some difference in the intensity of both of them, because when there is vibrational relaxation, that means the molecule is changing its conformation and it is using some kind of energy for that relaxation as well. So now what can happen is the light which it means absorbed, it is using some of its intensity here, some of its energy here. So now the light which is emitted, that intensity will be quite less as compared to the light which was absorbed. So this difference in wavelength, or in means the amount of energy which is absorbed, so this difference in wavelength is known as Stokes shift. Fluorescence occurs at lower energy, that means at longer wavelength. So I hope you are aware that there is an inverse relationship between energy and wavelength. So if energy is low, then that means wavelength is high, as we have seen in the case of radio waves, that they have longer wavelength but low energy. So fluorescence occurs at lower energy, longer wavelengths than absorption, due to the energy loss as the vibrational relaxation is undergoing in the excited state. And now this is the another characteristic transition times. So for the absorption, this is the time scale, that how much time is needed for the absorption, vibrational relaxation, and the lifetime of the excited state. Because this lifetime is also mattering, because when we were looking at in case of absorption spectra, when we were looking at the broadening of peaks, so we were also studying that if there is a difference in the time scale of that particular, means if a molecule is only remaining for a small amount of time in that particular state, then we also need some sensitive device which can capture that movement, because if time scale is very low, then sensitive devices are required. So this is the transition times for different types of transitions. So fluorescence is fast, which is 10 to the power minus 10 to 10 to the power minus 7 seconds. So which means 10 angstrom to 100 microseconds, while the phosphorescence is lower, that is 10 raised to minus 6 to 1 seconds. So intersystem crossing from S1 to T1, it takes longer than fluorescence, leading to delayed phosphorescence. And the fluorescence spectrum is typically a mirror image of absorption spectrum due to similar vibrational levels. So in the later slides, that will be more clear. So these different transitions we have already seen what intersystem crossing means, what vibrational relaxation means, what fluorescence means, what phosphorescence means. So this is just to show you that how much time it takes for each of these transitions, because if time is very less, then we require some sensitive device so that we can capture that particular transition. So now then another two terms. First is quantum yield and second thing is lifetime of excited state. Now you will see that why we were focusing on lifetime. So quantum yield is defined as the ratio of fluorescence photons emitted to the number of photons absorbed. So this will tell you how much yield is there. So photons emitted, that means in the fluorescence, there is some light which is emitted. And we have also seen by Stokes shift that due to the vibrational relaxation among these vibration levels, some kind of energy is absorbed. So the intensity of light of both of them means the number of photons. Intensity of light simply means that how many number of photons are there. So number of photons of absorbance will always be greater than the number of photons which are emitted. So in this case, quantum field will tell you that what is that difference. So it is the ratio. And you will always see that this quantum field will always be, means it will always be less than 1. And higher quantum field means there is a greater fluorescence efficiency. So in normal scenarios, if there is no such vibrational relaxation, then in such scenarios, quantum field will be 1. That means number of photons which are emitted will be equals to number of photons which are absorbed. But that is very rare because there is some kind of transition happening always. Then comes lifetime of excited state. Because when my electron is transitioning from S0 state to S1 state, this excited state, then it matters that for how much time it will stay in S1 state. So which is known as lifetime of excited state. That for how much time it will stay here. Because if it will stay here for longer time, then what it will be doing? It will be moving in these vibrational, it will showing vibrational relaxation and then it will come back to ground state. So for that reason, we need to see the lifetime. So average time a molecule remains in the excited state before returning to ground state. That is known as lifetime of excited state and it is calculated by the formula. So that tau is equal to 1 by eta plus KNR, where eta is the emissive rate, that means fluorescence, and KNR is the non-radiative decay rate, which means internal conversion or inter-system crossing. So these two scenarios are possible. So in such cases, these red lines are showing you that these are non-radiative transition. It is not radiating any energy, but some energy it is using and it is showing some transitions. So that's why the lifetime of excited state is very important to calculate to find out that what will be my intensity of this, how many number of photons it will emit in the fluorescence. Because if it will have this shorter lifespan or shorter lifetime of excited state, then that means in that case the fluorescence will be quite high because it is spending less time. So there will be less vibrational relaxation will be happening. So absorption excites a molecule to higher vibrational states. Then there will be non-radiative transitions, which will dissipate the energy before the fluorescence. And ultimately, fluorescence will move the molecule or move the electron from S1 state to S0 state by releasing some light of particular wavelength. Now we will talk about the instrumental setup for the fluorescence spectroscopy. So in this case, firstly, we have a light source, then we have excitation monochromator, we have our sample, and after the sample, we have the emission monochromator. So excitation monochromator is only allowing the fixed wavelength of light to pass through this particular sample. And now this sample is means absorbing the light, and then it is passing particular light through it. Now the emission monochromator is also allowing the wavelength of particular wavelength to pass through it. So depending on that particular wavelength, which has the higher, the wavelength which is greater, that will be selected from all the wavelengths which will be hitting the exit slit. So that particular wavelength will be detected by the detector. So in this way, it will tell you the difference among both the wavelengths and the means the increase, there will be increase in the wavelength because there is loss of energy. So this difference will tell you that how much light is absorbed or how much light is used for those relaxations. So this is the instrumental setup. Now we will look a little bit into the emission and excitation spectrum because fluorescence spectroscopy will give you this emission and excitation spectrum. So emission spectrum means the excitation wavelength is kept constant and the fluorescence intensity is measured as the function of wavelength. And this determines the spectrum of the emitted light. So emission means what is emitted, what is given out. That is known as emission spectrum. When we talk about excitation spectrum, it means the fluorescence intensity at a fixed emission wavelength is measured as a function of excitation wavelength. So in excitation spectrum, when we will be comparing it with respect to excitation wavelength is the wavelength of light, which is firstly hitting the electron and which is the energy which is gained by the electron to show the transition to excited state. So that is excitation spectrum. Now there are two possibilities, two spectral shifts which you might observe. First is red shift, which is also known as bathochromic shift, and this shift towards the higher wavelength. So bathochromic shift means that as the light passed, means as the light is absorbed and electron is excited, now when it will reach back to the ground state, it will emit some light. So the light which is emitted, it has higher wavelength. That means low energy. So some energy is lost. This particular shift is known as red shift. Whereas there is possibility you could also observe blue shift, which means hypsochromic shift. So which means shift towards the lower wavelength, higher energy and higher frequency. So which means if this wavelength is if the wavelength is reducing, so that means the energy which is emitted is higher, is of means the emitted wavelength or emitted light is high in means its energy is high and its wavelength is low. That particular shift is known as hypsochromic shift. Similarly, there are another shifts also. So for a particular wavelength, if there is increase in absorbance, then it is known as hypochromic shift, hyperchromic shift. And if there is decrease in the absorbance for a particular wavelength, then it is known as hypochromic shift, hypochromic effect. So hyper and hypochromic is based on the how the absorbance is changing, and red and blue shift is based on the how the wavelength is changing for a particular compound which has the same absorbance. Now we will see that what is the effect of solvent on the fluorescence spectra. So because we have seen that the fluorescence spectra is based on the principle of dipole moment and the excited state. So fluorophores have generally larger dipole moment in excited state than in the ground state. So this is also the reason that means if we use the fluorophores, then because of this large dipole moment in excited state, they will vary in the fluorescence spectra. Second thing is the solvent polarity and emission shift. So if there is polar solvents, then they will stabilize the excited state, leading to lower energy emission. That means they will show red shift. So for example, if I have an organic compound, then the wavelength was less. But as I increase the polarity, as I move towards the H2, which is water, which is polar in nature, so I'm increasing the polarity and this will also leads to increase in the wavelength. So this is the wavelength. So wavelengths are increasing. And we know there is inverse relationship between energy and wavelength. So high wavelength means low energy. So polarity or the polar solvents, they stabilize the excited state and leads to lower energy emissions and bringing about the redshift. Increasing the solvent polarity enhances this effect, shifting the fluorescence spectrum further to longer wavelengths. So this is also the reasons which affect the fluorescence spectra. Third thing is the pH sensitivity in the fluorescence. So now some fluorophores exhibit this pH dependent fluorescence spectra. This is due to an equilibrium between the protonated and deprotonated forms, which have different emission properties. So pH sensitivity is also important in fluorescence. This also influences the spectra. So these are the three reasons which can bring about change in your fluorescence spectra. Now we will look into the fluorescence quenching, because in fluorescence what we are looking at that means how much light is emitted. So it is based on the emission of light and also depending on what are the different vibrations, rotations are happening. So fluorescence quenching means the process which reduces the fluorescence intensity in a sample. And there are two types of quenching which we will see. Quenching means something which is means if something is reduced, that is known as quenching. So first is collisional quenching, and then we will also look at static quenching. So collisional quenching means it occurs when the excited fluorophores collides with the quencher molecule. So quencher is a molecule which is quenching the light which is emitted. So if there is collision between them, then this will facilitate the non-radiative transitions to the ground state, reducing the fluorescence. And common quenchers are oxygen and iodide acrylamide. Okay, now this is the representation that in fluorescence. you have the higher intensity of light. And in the case of quenched, the light is not emitted, so the wavelength is increasing and energy is decreasing. So intensity is low, that you can say. So now what happens in collisional quenching? There is collision. So in normal scenarios, you have a molecule, you have a luminophore which is absorbing some light, entering to excited state, again coming back to ground state by releasing some light, by releasing some uh, wavelength of light, which is known as fluorescence, that is coming from excited state to ground state. But what happened in the presence of quencher? So for example, if I have oxygen, which is a quencher, so light is absorbed, my molecule is excited to excited state. Now there is some quencher and my molecule collides with the quencher. So due to this collision, the energy is transferred. So whatever energy it has gained, it is transferred through this collision. It is lost in this collision. So it will come back to the ground state without emitting any light, any wavelength light. So this is known as quenching. And because in this case collision is happening, which is leading to non-radiative transmissions or non-radiative transitions. So that's why it is known as collisional quenching. And then there is equation given by Stern-Volmer, which says that F naught by F is equals to 1 plus KSVQ, where F naught is the fluorescence intensity without quencher, F is the fluorescence intensity with quencher, 1 plus KSV is the Stern-Volmer constant, and Q is the concentration of quencher. So which means if the concentration of quencher is high, then in that case, the fluorescence intensity will be as low. So the fluorescence intensity with quencher will be very low, because if quencher is strong, then it will not allow the means light. There will be no radiations that will be emitted. And then quenching rate, which is KSV, which is Stern-Volmer constant, it is equals to KQ tau 0, where KQ is the biomolecular quenching rate constant and tau 0 is the excited state lifetime without quencher. So this is the another relationship, tau naught over tau is equals to 1 plus KQ T naught Q, which means fluorescence lifetime decreases in the presence of quencher. Yeah, so this is about the collision quenching, and you only have to keep one thing in mind, that in this case, due to the collision, the means light is not emitted because there is non-radiative transition which is happening due to this collision. Now, second type of quenching is static quenching. So in the previous case, collision was happening and molecule was returning to its ground state. But in case of static quenching, what is happening is that my fluorophore, it is gaining some light, it is entering to its excited state. Now my means in static quenching, my fluorophore is coming. It is means the quencher is coming. Sorry, the quencher is coming and it will forming the complex with my fluorophore. So with this complex, now this complex is returning back to the ground state without emitting any light. But in this case, the complex is coming. In the previous case, the fluorophore is actually coming by itself. The quencher is separated, but in the static quenching, they are bonding together. And now both of them as a complex, they are coming to ground state. In this case also, there is no radiative transition. So no light is emitted in this case as well. And in this case, the fluorescence intensity can also be calculated by using the Stern-Volmer equation. So for this case, Stern-Volmer equation can be written as F naught by F, where F naught is the intensity without quencher, F is the intensity with quencher, which is equals to 1 plus KAQ, where K is the association constant. So means if there is a stronger associations, then the intensity will also be less, because if it will quench the fluorophore and it will not allow it to emit any radiations. So the key characteristic of static quenching is that it does not affect the fluorescence lifetime, which is tau, because uncomplexed fluorophores still emit normally, and it decreases the fluorescence intensity because by reducing the number of fluorophores available for emission. So that is the reason it is not reducing the lifetime, but it is reducing the number of fluorophores which were previously available to emit the light, but now because some are making complex with quencher, so intensity is decreased. And if we combine both of these quenching, for example, if there is collisional and static quenching both taking place, so in such scenarios, Stern-Volmer equation can be modified. It can be written as F naught by F, which is equals to product of both these uh, equations. So which is 1 plus KQ tau 0 Q and 1 plus KA Q. So in the previous case, we were also taking into consideration tau 0 because collisional quenching, they also influence the lifetime of the fluorescence molecule. But in this case, because it is independent of tau 0, so this is simply written as 1 plus KAQ. So it results in a non-linear Stern-Walmer plot. This is the Stern-Walmer plot. So in this, tau naught by tau is remain constant in the static quenching, whereas in this case, the slope is given uh, by this KQ tau 0. And in the presence of high temperature, this is means moving towards upper direction, whereas in this case, slope is decreasing. Slope is moving downward in the case of static quenching. So this is the difference among collisional quenching and static quenching, which is only happening if there is a quencher which is present. Now we will move to Foster's resonance energy transfer, which is FRET. So there are different energy transfer mechanisms which we have seen so far. So in this case, the fluorescence emission from a donor molecule is absorbed by an acceptor molecule. So there is this acceptor molecule which is absorbing the energy which is transmitted by the donor. So this transfer occurs only if the donor emission spectrum overlaps with the acceptor absorption spectrum. So in this case, both of their spectrum will be plotted. And if there is the overlap, then only this transition or this transfer can happen, in which the donor is transferring its energy to the acceptor molecule. So this is a non-radiative process. The FRET is a non-radiative process which does not involve photon emission. Instead, energy is transferred by the dipole-dipole interactions. So in all these cases, this is the modification of fluorescence spectroscopy. So in all these cases, we are seeing that it is based on the emission, that how much light is emitted. So there could be different mechanisms. So in quenching also, we have seen that it is a non-radiative emission because in this case, quencher is taking all that energy and the means molecule is returning to its ground state without any emission. Similarly, in the FRET also, what is happening is that the donor, when it is excited and reached to its excited state, then to come back to its normal state, instead of emitting the light, it is passing on this energy to the acceptor. So in this case, acceptor molecule is accepting that energy, but the precondition is that their distance should be less than 10 nanometers. And then only there will be some kind of overlap between their emission and excitation spectra. So this efficiency is depending on the distance between the donor and acceptor. And the equation is E is equals to R raised to power minus 6 divided by R raised to power minus 6 plus R naught minus 6, which means that R is the distance between the donor and acceptor, and R naught is the Foster distance, where the FRET efficiency is 50%. So ultimately, FRET tells you that whether there will be transfer happening or not. So depending on which, you will see there will be difference in the absorption and emission spectra with respect to wavelength. So there is, first of all, there is distance dependence. So distance dependence means if there is distance less than 10 nanometers, then there will be high energy transfer efficiency. And if the distance is greater than 10 nanometers, then the energy transfer will not take place. So in such scenarios, there is no energy transfer. Second dependence is the angular dependence. So it also depends that whether what is the orientation of donor molecule and the acceptor molecule. So FRET will only occur if their dipole alignment is not perpendicular, because this transition is happening based on the dipole-dipole interaction. So if both the dipoles they are means if they are not perpendicular to each other, then only this transfer can happen. And if angle is equals to 90 degree, then no FRET is observed. So these are two preconditions which are means if they are satisfied, then only the FRET is happening. That is Foster's resonance energy transfer. Now we will talk a little bit about the fluorophores in proteins, because the fluorophores are the molecules which emits the light in the form of, and showing the fluorescence. That is the molecules which we are observing in the fluorescence spectroscopy. So there are some molecules which are known as intrinsic fluorophores, such as tryptophan, tyrosine, and phenylalanines, because the fluorescence will only be shown by those molecules or those amino acids which can absorb the light and which can further show excitation and then emission. So in this case, proteins have these common fluorophores, in which the tryptophan was the strongest because it has highest absorption. And we have also studied that it has the conjugated system, due to which its absorbance was maximum. So if it absorbs the maximum light, then its ability to emit the light will also be high. Similarly, tryptophan and tyrosine, they produce the stronger fluorescence as compared to phenylalanine, and tryptophan is often quenched due to the proton transfer from excited state. So it is the dominant fluorophore. And then this tryptophan fluorescence, it is also sensitive to environment. So due to which we can study conformational changes, subunit association, substrate binding, denaturation. So all these things can be studied by using the fluorescence spectroscopy. And these are the plots which are telling you that excitation and emission spectra is plotted. So we know that there is some stock shift which is happening because we know that there is some vibrational relaxation which is also taking place. So the amount of energy which is absorbed, so the light is not emitted with the same energy. There is some loss of energy due to which there is increase in the wavelength of light which is emitted. So that's why in absorption spectra we'll always have lower wavelength, whereas my emission spectra will always have the higher wavelength. So this is what the plot is showing, and these are some of the different features of different fluorophores, such as their lifetime, absorption, absorptivity, fluorescence, quantum yield. So these are some of the parameters which you can go through. Now there is another fluorescent protein which is known as green fluorescent protein, which is most commonly used. So it is composed of 238 amino acids, and it is a monomer structure which has a central alpha helix, and it is surrounded by the 11-stranded anti-parallel beta sheath cylinder. So there is one circular alpha helix which is surrounded by these beta sheath cylinders. Its diameter is about 30 angstrom and its length is about 40 angstroms. And these fluorophores, they are embedded within the central alpha helix. So central alpha helix have those molecules which are involved in the absorption of light and showing the fluorescence. So how this fluorophore is formed? So there is a sequence which is observed in them. So this chromophore sequences serine 65, tyrosine 66, and glycine 67. So this particular sequence is responsible for the fluorescence to happen. So there is a maturation mechanism. So this is how the green fluorescent protein is synthesized. Firstly, there is cyclization of the extended polypeptide backbone, and then imidazolinone ring system formation is happening. Then after dehydration, cyclized ring system is formed, and after its oxidation, we ultimately obtain the green fluorescent protein. So you don't have to worry about it. You only have to keep one thing in mind, that the fluorescence molecules, they have some. So for example, in this case of GFP, they have some characteristic sequence which is present, or you can say characteristic means uh, yeah, means recognizing this stretch of amino acid which is present, which is responsible for the fluorescence phenomena. So that is serine 65, tyrosine 66, and glycine 67. And these 65, 66, and 67 represent the positions because this is 238 amino acid long. So at position 65, 66, and 67, it has serine, tyrosine, and glycine. But now, after performing all the experiments, it was later on realized that if we bring about some mutations in this particular sequence, then we can change the color which it emits and we can also enhance the signal. So this green fluorescent protein, it shows the green light because it emits the green light, or the means the wavelength of light which it emits, that is in the green color. So that's why we call it as green fluorescent protein, because it appears green. So similarly, if we bring about the modification in this particular sequence, then we can decide. So in case of EBFP, which is the enhanced blue fluorescent protein, if we modify the 66th position, so in place of tyrosine, if we add histidine, so then in that case, instead of emitting green color, it will emit blue color. If we made the modification at the 65th position, which is instead of serine, we add threonine. So in such case, we can enhance this intensity of GFP. So that is known as enhanced green fluorescent protein. So efficiency is enhanced. Similarly, if at the 66th position, instead of tyrosine, we add tryptophan. So in such scenarios, it will give the cyan color. So that will form an enhanced cyan fluorescent protein. Similarly, for yellow color, we can change the position at 65th. Instead of serine, we can add glycine. And at 203rd position, we can add tyrosine. So that can also increase the intensity of the yellow light. So it will emit the light which is of wavelength equals to the yellow light. So it is known as the enhanced yellow fluorescent protein. So this shows you that what is the composition of fluorescent molecule or fluorescent protein, and how with the modifications we can even vary that what light it will emit. So this is how, if we have different proteins, then their spectra can vary depending on the what type of amino acids are present. And in all these cases, keep one thing in mind, that we are not changing the aromatic group. So in the chromophore sequence, there must be at least one amino acid which is aromatic. If there is no aromatic amino acid, then it will not show fluorescence. So in all these modifications, if we are changing the tryptophan, then we are only changing it to the another aromatic group. So we are not just changing it to any other random amino acids. So this thing has to be kept in mind. And now we will move to infrared spectroscopy. So why do we use infrared spectroscopy? So this studies the absorption of infrared radiations, and this causes the vibrational transitions in molecules, and it is used for the functional group identification and structural elucidation. And most of the times when we are performing IR spectroscopy, then we generally explore the mid IR region. So there are three regions. One is near IR, which is from zero point 0.8 to 3 micrometer. Mid IR, 3 to 8 micrometer, which is used for most analytical applications. And then fair IR, which is from 8 to 15 micrometer. So the bond vibrations, because it is based on these vibrations. So covalent bonds, they are not rigid. They behave like springs, and they can vibrate when they absorb some energy. So you can see that till now, whatever spectroscopic techniques we have seen, there is some kind of changes which are happening due to which we are able to study. So in case of infrared, there is some vibration which is ongoing. So there are two main types of vibrations which we observe in infrared spectroscopy. So first is stretching. Stretching is a type of vibration in which there is change in the bond length. So it could be symmetrical stretching, in which from both the directions we are moving the atoms in the same direction. We are stretching them in the same direction. That means either we are increasing this bond length from both the sides, or we are decreasing this bond length. Whereas asymmetrical stretching means that if from one side we are pulling the atom, pulling the bond, then if from the other side we are pushing it inside. So in this case, the bond length will vary from both the sides. So for example, if it is H2O and these two are two hydrogen atoms and this is the oxygen, then in this case, the bond length of this OH group and this OH will vary. One will be large, one will be short in case of asymmetric, whereas it will be constant in case of symmetrical stretching. Then comes bending. So bending comes into play when there is change in the bond angle, and there is bond length remains same, but bond angle is changing. So for example, similar example, if I take this is oxygen and these are two hydrogens, then in case of in-plane bending or rocking, what I am doing is I am moving them in opposite directions, in other directions. So in this case, there will be change in the bond angle. In case of scissoring, if both of them means it behaves like a scissor. So in this case, either they both can come close to each other or both can go far from each other. That is known as scissoring. But this type of bending is known as in-plane bending. That means they are in the same plane. They are just moving either closer or farther from each other. Whereas in case of out-of-plane bending, which means they are moving up and down. So for example, in twisting, if one means one hydrogen, one means this is oxygen and these are two hydrogen. If, for example, then one of the hydrogen we are means pulling it towards upside, whereas we are bending this another hydrogen downside. So in this way also, I am changing the angle which is formed between these two bonds, and we are moving some one up and one down. That is known as twisting. Then the another is wagging, which means at the same time I am moving both of them upside. So that means the direction of movement is same. Either it is upside for both or either it is downside for both, which is known as bagging. This comes under bending. So in infrared spectroscopy, we generally explore these types of vibrations which happens in the bond. And stretching frequencies are most useful in case of IR spectra analysis. So this kind of stretching is more important. And in IR spectroscopy also, we have two spectrum. One is transmittance and another one is absorbance. So both of them, they are symmetrical to each other. So in the places where there is absorbance, so the transmittance will be low. And if transmittance is 100%, that means absorbance will be zero. So IR spectroscopy, it measures the absorption and the transmission of IR light by the chemical samples. And IR spectrum is a plot of frequency versus intensity. So in this case, this is the wave number and this is the absorbance and transmittance. So this can also be represented as the frequency as well as intensity. And absorbance spectrum, it displays the absorbed frequencies, whereas this transmittance or transmission spectra, it tells you the how much light is transmitted. And then you can also classify these IR bands into three categories. First is strong, which means if there is high intensity. Then comes medium, which is moderate intensity, and then low intensity. So because if we consider it as to be intensity and this to be the x-axis to be the frequency, then this is how the strong band appears, medium bands and weak bands. Now we will talk about the IR active bonds, because if a molecule has the IR active bonds, then only we'll be able to get that spectrum. So only the polar covalent bonds absorb the IR radiations and appear in the IR spectrum. So this is one of the conditions, that there should be polar covalent bond. Band intensity, it's depending on the dipole moment strength. So strong band will be only appeared if there is a higher polar bonds such as carbonyls. Medium bands is observed for the moderately polar and asymmetric bonds, whereas weak and no bonds if I have some compound which has a weakly polar or symmetric bonds. So in case of symmetric also, because the dipole moment will cancel out each other, so I won't be able to observe the IR spectra, so that will be IR inactive. So symmetrical bonds, no dipole moment change, IR inactive. Asymmetrical bonds, dipole moment change, and they are IR active. And signal types can also be classified as broad signals or narrow signals. So when you observe the broad signals, so due to hydrogen bonding or if there are complex vibrations, so in such scenarios, there could be broad signals being observed, and sharp peaks are observed from the isolated bonds. So in case of carbonyl group, aldehyde, ketone, you will see a strong IR band, whereas in the nitriles, such as C triple bond N, you'll see a medium bond, medium IR spectra or medium band. In case of this alkynes, you'll see there is a weak band. So yeah, weak band which you will observe for the IR spectra. Yeah, so this is how the narrow signal and the narrow signal and a broad signal appears. This is just the representation, and this is also to show you that what are IR active and what is IR inactive, because in IR active dipole moment cancel out each other because there is symmetric stretching. If there is asymmetric stretching, dipole moment will be present, it will vary among both the poles. So change in dipole moment make it IR active. And this is the characteristic IR absorption values for different types of bonds. And from this, keep a few things in mind, that the groups such as C double bond O, C triple bond N, C, so they have, this is the distribution that which has the strongest IR band and which has the weakest IR band. And this is another example that how you can classify among different types of compounds. So this is for the distribution. So in this case, we have considered aldehyde, this is the ketone, and this is the acid, COOH, which contains COS, carboxylic acids. So they are the different compounds, and this is their means this is the main category. So in case of aldehydes, you will see two different peaks. One for this strong CO peak, CO stretch means there will be the stretching of this C double bond O, which will give you the peak at this particular wave number, which is from 17 means 1710 to 1720 centimeter inverse, due to the high polarity of C double bond N, C double bond O. And in aldehydes, we also have the additional CH bond. So this CH bond will also show this weak band, which will be at 2700 to 2800 centimeter inverse. This is the wave number, and this is absent in ketones. So in both the aldehydes and ketones, this CO band will be visible, but this CH band will only be observed in case of aldehydes. So this is how we can distinguish among the aldehydes and ketones if we have two samples. Now comes carboxylic acids. So in carboxylic acids also, we will see this C double bond O stretching, which will appear in the at the wave number 1710. But the main important unique feature of this carboxylic acid is that there is OH group which is present. So this OH stretching, it will be a broad band which is observed around at the wave number of 2800 to 3500 centimeter inverse. So this shows that all of these three compounds, they have characteristic IR spectra due to which we can distinguish among them because they have different bonds and they show different stretchings. I hope it is clear till now. Now we will see IR spectroscopy of amines and amides because in proteins, these amide bonds are present. So in case of means amines in which the NH bond is present, so primary amines have two NH bonds because in this there is NH2. That means there are two bonds between NH and H. So two bonds are present. So there will be two spikes that you will observe. In case of secondary amines, because there is only one band that is NH, so only one spike is observed. In case of tertiary amines, because there is no H which is present, so NH is not there, that bond is missing, so no IR band will be observed in the tertiary amines. Now if we talk about amides, so in this case, um, primary amides, they will also show two spikes due to the two NH bonds which are present. And similarly, in the case of secondary amine also, because one H is present, so one NH bond is there, so there will be only one spike, which is due to the one NH bond. So with the help of this IR spectra, you can also identify that the compound which you are observing, whether it has the amines, it has the amides, or it has the primary amides or secondary amines, depending on the how the peaks or how the spikes are observed. If two spikes, one spike. So this tells you that how the IR spectra is helping you to distinguish among two compounds. Now there are certain applications. First of all, detection of solvents. So standard IR spectra of common solvents, this is the different common solvents IR spectra. So now if you observe the spectra of unknown compound, then you can compare them and you will be able to know that what is their unknown solvent. Second thing is detection of unknown sample. Similarly, you can also compare the unknown spectrum with the known reference sequence and you can find out, you can predict that what could be that unknown sample if it matches with the already known spectrum. Then comes air contamination analysis. So with the help of IR spectroscopy, you can also quantify the air pollutants such as carbon monoxide, methanol, ethylene oxide, because these compounds have different bonds and different bonds have different dipole moments. So due to different dipole moments, they will show different vibrations, or you can say they can show different stretchings. So due to which you can distinguish their spectra among one another. So in this way, you can identify that what is the contaminant in the air which is present. Along with this, you can also identify their concentration because these peaks will also tell you about the concentration. Yeah, so this is all about the IR spectroscopy. And then comes the Fourier transform IR spectroscopy, which is the modification of IR in which we are increasing the intensity of signal which we are observing by performing Fourier transformation. So it collects the high resolution IR spectra of samples absorption and emission over a wide range. And it uses the Fourier transform to convert the raw data into the actual spectrum. And what are the application of this FTIR? So it provides the secondary structure information such as alpha helix, beta sheets. And unlike X-rays, X-ray crystallography, NMR, it focuses on the structural content rather than the tertiary structure. So it is based on the secondary structural content and key IR bands in proteins. So because we know in proteins there are amide bonds which are present. So in this case, for different proteins, we will observe characteristic amide 1 and amide 2 bands. So amide band generally appears around from 1600 to 1700 centimeter inverse, and amide 2 band is observed from 1500 to 1600 centimeter inverse. So this is the wave number. Other notable stretches are CH, PO, and amide A bands. And in this plot also you can see that for different types of secondary structure, we have different values of different wave numbers. So with this, with the use of Fourier transform infrared spectroscopy, you can also identify that what type of secondary structure is present. And it is enhancing the signal intensity from IR. So that's why it is more commonly used. And in case of proteins, we focus on the amide 1 and amide 2 bands. Now we will move to our last scattering or last spectroscopic technique, which is the Raman spectroscopy. And because Raman spectroscopy is based on the scattering of light, So that's why it is known as Raman scattering. And C.V. Raman was the one who was the first Indian to get the Nobel Prize in physics in 1930s for this Raman scattering. So what he said, that scattering is a change in the direction of particle due to its collision. So Raman scattering is, it says that a small fraction of scattered light shifts in the frequency due to the molecular vibrations, and it provides insights into the chemical composition and molecular structure, because collision can only happen, means it depends on what molecules are present in the neighborhood. So that will influence the collision, and depending on that collision, means the wavelength of light can change. So the Raman effect is weak, and this effect is very weak. So to check or to compare or to identify this effect, we require a very sensitive instrument, because this effect is very weak, such weak that 1 in 10 million photons they shift the frequency. So it's very hard to identify them. So types of scattering. Scattering can be three types. First one is Rayleigh scattering, which is known as elastic scattering, which says that the light which strikes the molecule, the same light is emitted. So that is known as Rayleigh scattering. That means the wavelength of the scatter is equal to wavelength of laser. And also keep one thing in mind, then that in all other techniques we are using some kind of light source, but in Raman scattering we are using the light which is laser light. So in this case, the fraction means so the wavelength of laser is equals to the wavelength which is scattered. Then comes Stokes Raman scattering. So Raman told two types of scattering. One is Stokes Raman scattering and anti-Stokes Raman scattering. Because this was very weak, but still he observed. That's why he got the Nobel Prize. So what he said, that the scattering is not always elastic. There is the inelastic scattering is also possible. So in some scenarios, what can happen is there is some loss of energy due to the collision. So in that case, the wavelength of scatter will be more than the wavelength of laser. And I'm again saying the same thing, that when you say increase in wavelength, which means there is decrease in energy. So due to collision, some energy is lost. So the energy which is emitted is less. That means it has the greater wavelength. So which is told about by the Stokes Raman scattering. That means the wavelength of scatter is more than the wavelength of laser. Then there is another type of scattering which can also be observed, which is known as anti-Stokes Raman scattering, which means scattering is inelastic. That means there is change in the wavelength. So energy is gained. So in this case, the wavelength of scatter is less than the wavelength of laser. That means it has high energy. And the application of Raman spectroscopy is to study the vibrational, rotational, and low frequency modes in materials. Yeah, so this is the representation of anti-Stokes, Stokes, and Rayleigh scattering. In this anti-Stokes and Stokes scattering, so they are shown by very less number of photons. And yeah, so this is just the representation that how the transition is happening among different energy states. So these are the vibration energy states which are for the same energy level. There could be multiple vibration states which are possible. So generally what is happening is that the molecules, it absorbs the light, it excites to the excited state, and then it comes back to the means to the ground level. But in case of inelastic, what is happening is that because the some energy is lost in the collision. So in case of Stokes Raman scattering, the wavelength of light which it emits is less. That means it, instead of reaching to the ground state and the last vibrational level, it is reaching to some above level. So not the last vibrational level, but to the first vibrational level. So this particular gap which is there, this is the difference in energy which is lost in the collision. So that's why the wavelength is increased in case of Stokes Raman scattering. Whereas in case of anti-Stokes, so in this case, if transition was happened from the first vibration level, then because gain of energy is taking place, so it will reach to the means last vibration level, that is level 0 of the ground state. So that means there is this extra energy is gained. So this decreases the wavelength. Okay, now we will compare between IR and Raman spectroscopy. So in IR, we have seen that it was based on the absorption of light and there was vibrating molecules. Peaks correspond to changes in the dipole moment, and it operates in the infrared region of the EM spectrum and most probably the mid IR region. Whereas if we talk about the Raman spectroscopy, then it is based on the scattering of light. It is not based on the absorption but scattering by the vibrating molecules, and its peaks corresponds to the changes in the polarizability, and it works in the visible near IR or near UV regions, And now there is the mutual exclusion principle, which says that the molecules with the center of symmetry, so IR active vibrations are not Raman active, which means there is a mutual exclusion between both of them. So if a molecule shows IR uh, spectroscopy, if a molecule is IR active, then it will be Raman inactive and vice versa. So this is the example. So these are the vibration modes. For example, this is the equilibrium structure. This is the molecule, and it is showing the means it is the equilibrium structure which is most stable. So there is no activity at equilibrium positions because it will not show vibration anything. It is the most stable state. So it is Raman and IR inactive. If there is symmetric stretching, which means there is change in bond length from both the direction, this bond length is equally changed. So that means there is no change in the dipole moment. So if there is no change in net dipole moment, then it is IR inactive. Whereas it will be Raman active. So in this case, it is Raman active. Then comes asymmetric stretching. So if there is asymmetric stretching, so in such scenarios, there is change in dipole moment. So dipole moment don't cancel out each other. So that's why it is IR active. Whereas if we find the ratio of them, then it comes out to be zero. So it is Raman inactive. Then comes bending. So in case of bending, it is IR active. Whereas in case if there is this particular type of bending, so in this case it is Raman inactive. So just keep one thing in mind that the symmetrical stretching which was IR inactive that can be studied using the Raman spectroscopy because it is Raman active. So these are the advantages of Raman over IR. And in other advantages are that the water can be used as a solvent, which is ideal for the biological samples. So Raman scattering can be performed by using the molecules in the water sample as well. And symmetric vibrations are also observable. And Raman intensity is proportional to the concentration and laser power, because in this we are using the laser light. These are some of the advantages of Raman spectroscopy. And now this Raman spectroscopy has two applications. So there are two types of Raman spectroscopy. One is resonance Raman spectroscopy, in which the excitation wavelength is chosen to match or be very close to the electronic transition, usually in the UV visible range. So this resonance enhances the Raman scattering intensity significantly, and it allows the detection of compound at very low concentration. Scattering intensities increase by the factor of 10 raised to power 2 to 10 raised to power 6, which reduce the detection limit and measurement time. So this is known as resonance Raman spectroscopy, where you are deciding that what should be the excitation wavelength. Then comes surface enhanced Raman spectroscopy. So in this case, you enhance the Raman scattering when the molecule are adsorbed on the rough metal surface. So there are some molecules which are further adsorbed on the surface, and then those adsorbed molecules are enhancing the intensity. So this enhancement factor is as high as 10 raised to power 10 to 10 raised to power 11. And this enables the detection of single molecules. So these are some types with which we can increase the intensity, because we have seen that anti-Stokes shift, so the two types of Raman scattering, so they are shown by very less number of photons, 1 in 1,000 photons. So their intensity is low. So we can increase this intensity so that detection rate can be enhanced. So these are two types of Raman spectroscopy with which we are increasing the intensity of our signal. Now Raman spectroscopy can be used for the proteins. So secondary structure analysis can be done. In this also we get amide 1 band and amide 3 band. So the major contribution is by the C double bond O stretch vibrations, and it is located around the 1650 1650 centimeter inverse. And different secondary structures such as alpha helices, beta sheets, random coils, they exhibit variation in the position and shape of this amide band, due to which we can distinguish among both all these secondary structures. Similarly, amide 3 band, it arises from the CN stretch and NH bending vibrations. So NH bending vibration contributes 30% and CN stretching contributes the 40%, and it is found at around 1300 centimeter inverse. And different secondary structures, alpha helix, beta sheet, random coils, they show distinct peaks in this amide 3 region as well. So based on this particular peaks, you can distinguish that what is the secondary structure present in the protein which you are studying. So with this, Raman spectroscopy because it allows you to study the samples in the liquid. So you can study the dynamics, that how by adding some molecules or by adding other compounds or by increasing the temperature, how the secondary structure components are changing. So this is how the spectroscopy can tell you. So, but these techniques are based on the secondary structure, and they're not focusing that what is the tertiary structure which is present of the particular protein. And that tertiary structure is obtained from X-ray crystallography and NMR. So that's why those techniques are majorly used and not these. These only tells you at the level of secondary structure. Then another applications of Raman spectroscopy are Raman microscopy and Raman crystallography. So in Raman microscopy, there is just one addition of a confocal microscope to the Raman spectrometer. And why it is helpful? Because by using the confocal microscope, the one thing you are doing is you are completely focusing on the sample. So you know from where or which part to excite, where the light will be striking on my sample. So in this case, even if my sample concentration is low, I will be very precise enough to pass the laser light through it. So advantage is it that it provides the high magnification visualization, and it also enables the analysis at different spatial resolutions, because there is two types of mirrors which are present. So in this case, um, there is the this di there is a dichroic filter which is present, so which allows that the light will be, when it is falling on the sample, then at different positions, at different points, the light will fall, and at each point, the means after this light is falling on that point, that is detected by the detector, so it can combine all those, from all those points, the complete image can be formed by looking at the spectra. So that's why it uses the spatial filter, which is known as pinhole, to ensure that the controlled sampling volume is controlled. And it also enhances the depth selectivity by filtering out the out of focus Raman scattering. So not all the scattered lights are captured. So filtered out the Raman means out of focus lights, and it allows the 3D Raman imaging. So with the help of this, we can also perform the 3D Raman imaging. And the basic setup is that you use the laser which excites the sample. So from this laser, the um, light is hitting the sample, and then from the, because you are focusing it with the confocal microscope, you are focusing, you are magnifying the sample, and then you are passing the laser. That laser light will be reflected back by this, and it will be captured, means there is some kind of uh, change in the intensity of light which is further means which is scattered by the sample, and that scattered light is captured, and then another lens is present and other filters are present which make sure that the out of phase Raman scattered lights, they will not be counted, and the proper in phase light will be uh, allowed to pass through this filter. And later on, the spectrometer is present, which will check for the intensity of that light and other calculations that will perform. So in this case, generally it finds out the Raman shift, that how much change in the wavelength is happening. So that is known as Raman shift. So in the ultimately, it will give you the plot, that how the absorbance versus the Raman shift. Okay, so these are some of the components. And now last thing is the Raman crystallography. So in Raman crystallography, you are studying the sample in which you are studying the crystal. So that's why it is called as crystallography, because you are studying the protein crystal. And in this case, it is in the form of hanging drop. And then the laser light is passing through that hanging drop crystal. So one of the advantage of this particular Raman crystallography is that it has the high signal to noise ratio with non-resonance Raman conditions, and there is low background noise because there is minimal luminescence, and it also provides the excellent platform for Raman different spectroscopy. And in this case, we can also study the protein ligand interactions. So in that case, in the crystal only, we can also add the ligand and we can co-crystallize the protein and ligand, and then we can study the Raman, means we can use the Raman spectroscopy and we can see that how the interaction is happening live. So this is why we use the Raman crystallography. The major difference is that in this case, we are studying the crystal. So if protein is studied in its crystal form, then that's why it is called as Raman crystallography. And it helps to track the chemical changes in the single protein crystal, which complements the X-ray crystallography. Yeah, so these are the advancements of Raman spectroscopy, microscopy, as well as crystallography. Now we will quickly move to the practice questions. and we will go through them. Okay, so now let's talk about the first question, which is the selectivity and sensitivity of fluorescence spectroscopy is higher than that of the absorption spectroscopy because of all of the following except. So option A is fluorescent substances have different lambda max of excitation and emission. Option B is rigid molecules can only fluoresce. Option C is fluorescence intensity depends on the intensity of incident radiation. And option D is every molecule that absorbs in the UV region can show the fluorescence. Okay, so anyone wants to answer? Okay, now let's see what is the right answer. So the right answer is option D. That is, every molecule that absorbs in UV region can show fluorescence. So in option A, it says that the fluorescent substance have different lambda max of excitation and emission. So this is true because we have seen that in the case of Stokes shift, so which enhances the specific selectivity, because we have seen that the lambda values of excitation and emission, they vary, due to which we explore this. Second option was that the rigid molecules can only fluoresce. So this was also true because the rigidity reduces the non-radiative decay, which happens when the relaxation is happening in the vibrational level. So because the rigidity will reduce the non-radiative decay, so this will increase the fluorescence efficiency. Third option was that the fluorescence intensity depends on the intensity of the incident radiation. So this is also true because if the intensity which is hitting the molecule, if the incident radiation, if that is having high intensity, then there is a possibility that the fluorescence will also have the higher intensity. If your intensity of absorption is only low, then how can you expect that the emission can happen at the higher intensity? So that's why there is proportionality, and fluorescence intensity depends on the incident radiation intensity. And option D was that every molecule that absorbs in the UV region can show the fluorescence. So this is false because many molecules they lose the energy through non-radiative pathways instead of fluorescing. Because we have seen that if there is a quencher molecule that is present, then there is a possibility that it does not fluoresce. Now the question number 2, the transition zone for the Raman spectra is option A between electronic levels, option B between vibrational and rotational levels, option C between magnetic levels of nuclei, and option D between magnetic levels of unpaired electrons. So this Raman spectra we have just discussed that what are the transition zones. So in this you have to focus on that what are the changes that are happening in the atom due to which we are able to distinguish among different types of Raman spectra for different secondary structures or other things. Okay, so now let's see what is the right answer. So the right answer is between the vibrational and rotational levels. So these are the two transition zones which are happening. Yeah, so this is just the representation we have discussed that how the vibrational and rotational energy levels are changed and which can means due to which the Stokes shift, anti-Stokes shift, so that is observed. Stokes Raman scattering, anti-Stokes Raman scattering. So this is based on the vibrational and rotational levels. Now question number 3 is which amino acid in green fluorescent protein is not essential for the formation of chromophore and green fluorescence? So option A is serine, option B is glutamine, option C is tyrosine, and option D is glycine. So when we were talking about green fluorescent protein, then we have seen that there are three characteristic amino acids which forms the chromophore. And by modifying them, we can enhance the intensity of GFP or other things, but those three amino acids were fixed. So now you have to tell the one which is not essential. Okay, now let's see. So the right answer is glutamine because the three amino acids which are important were serine, tyrosine, and glycine, which we have seen here. Serine at position 65, tyrosine at position 66, and glycine at position 67. Next question is chemical events within the crystal can be obtained from which of the following techniques? X-ray crystallography, NMR, Raman spectroscopy, and Raman crystallography. Yeah, so this is also quite straightforward. So the right answer is Raman crystallography. And in order to avoid the confusion between the X-ray crystallography is that in this they are saying the chemical events within the crystal. So in case of X-ray crystallography, there are not much movement of the protein molecule inside the crystal. It was more like, it's more like rigid. It does not have that much means freedom to change the conformations or undergo interactions. So that's why in the Raman crystallography, because it is based on the hanging drop method, so in this case, we can study the chemical events. We can study the interactions very easily, which are not that easy in the case of X-ray crystallography. Okay, next question is identify the correct decreasing order of wavelength in the electromagnetic spectrum. So this is the option A, B, C, and D. And by this time, we have seen the electromagnetic spectra by a lot of times. So this will be quite easy. Okay, so first of all, before looking the answer, first thing that if you don't know the answer, then you can cancel out those options which can not be true. So in this case, they are saying decreasing order of wavelength, that when we move forward, then the wavelength should decrease. And I've also told you so many times that increase in wavelength means decrease in energy. So decrease in wavelength means energy increase. So that means if we move from left to right, then energy should increase and wavelength should decrease. And we have seen in the previous class that radio waves have the lowest energy. So in this case, A and B options are cancelled out. Now in this case, the second option is ultraviolet versus visible. So now in this case, you have to figure out that whether the UV rays have high energy or visible rays have high energy. So by cancelling out this, you will be able to know that the right option is option D, because infrared has the lower energy and high wavelength than ultraviolet. After infrared, we see visible and then UV and then X-rays. So this is the electromagnetic spectrum, and this is how you can remember it. And also focus that what the question is asking. It is asking you decreasing order, increasing order, or what. Okay, so now let's move to question number 6, which is Beer-Lambert's law states that option A, absorbance of light is directly proportional to sample concentration. Option B, absorbance of light is directly, or you can say is inversely proportional to sample concentration. Option C is sample density is directly proportional to sample concentration. And option D, none of the above. Okay, now let's see. So the right answer is the absorbance of light is directly proportional to sample concentration. So this by Lambert's law, we have seen that if the concentration of sample is high, that means greater number of solute molecules that are present. So if solute molecules are more, then they have the ability to absorb more light. So that's why there is a straight line plot between absorbance and concentration. Because with increase in concentration, the absorbance is also increasing. So the light which is incident and the light which is transmitted, it will vary in the wavelength or energy, because the concentration is high, so transmitted light have low intensity than the incident light. Next question is molar extinction coefficient of a molecule is dependent on. So in the previous equation where we have seen this epsilon, in some cases you might see it as a small a. So this is known as molar absorptivity or molar extinction coefficient. So in this question, they have asked that the molar extinction coefficient of a molecule is dependent on option A, density, option B, viscosity, option C, concentration, and option D is wavelength, that is lambda. Okay, so now let's see. So the right answer is wavelength. So this molar extinction coefficient, it depends on the wavelength. Different molecules absorb light differently at different wavelengths, because it tells you that what is the absorption of a particular molecule. So it does not changes with concentration, or because it's the ability of the molecule. If you are increasing its concentration, there is no change in its absorptivity. Its ability is the same. So it changes with the wavelength. So that's why the right answer is option D. Next question is which two signature negative positive bands can one observe when performing a CD spectra of beta pleated sheet in the protein? Option A is 216 nanometer and 196 nanometers. Option B, 220 nanometers and 194 nanometers. Option C, 218 nanometers and 195 nanometers. And option D is 217 nanometers and 197 nanometers. Okay, so in this case also, when we were looking at the plot, for polyl lysine, then we have seen that for the case of beta pleated sheet, which is represented in the orange color, we see two bands. One is the positive band, another one is the negative band. So positive band is observed at 195 nanometers and negative band is observed at the 218 nanometer. So this is the reference CD spectra, which is generally kept in mind while comparing it with other spectra, and then we find out that what kind of secondary structure is present in our sample molecule. Yeah, so the option C is the right. Now next question is Raman microscopy, also known as micro Raman, is option A chemical analysis technique, option B is a Raman spectroscopy, option C is a traditional light microscope, and option D is the combination of all these three. Okay, so the right answer is combination of all these three. It is quite confusing, but yeah, we know that it is a chemical analysis technique. We have seen that it is the analytical method. Second thing, the Raman microscopy is the application of Raman spectroscopy. So it is a Raman spectroscopy. Now the next thing is traditional light microscope. So in generally we use the confocal microscope. So in this case, because these two options appear to be more correct, so instead of selecting one option, we can select combination of all these three because there is some light microscope which is used, specifically confocal in case of Raman microscopy, and this is how the instrumental setup of Raman microscopy, and this is how the originally instruments look like. So in addition to Raman spectrometer, it has the confocal microscope which is attached, due to which we can magnify the sample through which we are passing the laser light. Now the last question is circular dichroism spectroscopy is option A sensitive to the primary structure of proteins, option B sensitive to the secondary structure of the proteins, option C sensitive to the tertiary structure of the proteins, and option D sensitive to the quaternary structure of the proteins. Yeah, so this is also we have discussed that circular dichroism, it is sensitive to the secondary structure of the proteins. And till now, most of the spectroscopic techniques which we have seen so far, they are sensitive to the secondary structure level only. None of them gives us the sensitivity at the level of tertiary structures. That's why we most commonly use the X-ray crystallography and NMR for the tertiary structure. And in later sessions, we'll see that how the cryo-EM is also used. Yeah, so this is the in this particular figure to show you that what a primary structure, secondary structure means. So in secondary structure, we mainly focus on the alpha helix, beta sheets, turns, and random coils. And there are hydrogen bondings which are present. And in this case, each type of secondary structure give a characteristic peak. So due to which we are able to distinguish among them that what kind of secondary structure is present. So by looking at the CD spectrum, we can predict the secondary structure, that what secondary structure my protein can adopt based on the CD spectrum, and ultimately we can also predict that what fold it can take because different secondary structures combine to form the super secondary structures which are ultimately motifs. So means by combining all of them, we can predict the fold that my protein can adopt. So this is all about today's session. And in the next session, we'll be talking in detail about the microscopic techniques. So if you have any doubts related to the spectroscopy, then you can ask. Or otherwise, you can also ask in the discussion forums if you don't have any doubts means right now. So I'll be answering from discussion forums also. And even if you're not able to ask your question in discussion forums, then you can also comment in the video, which means because these videos are recorded and this will be uploaded in the YouTube. So means you can also comment in each video if you find any doubt at any point of time. Okay, then I'll end the session. Thank you very much. We'll meet in the next session.